Hey everyone, today we are talking about blood, and hopefully you've already had some labs on the cardiovascular system, including blood. So some of this information should feel a bit familiar, and that's going to be great. Just don't mistake familiarity with the topic as the same thing as actually knowing the topic for a unit test, for example. Okay, let's remind ourselves that the functions of blood are actually quite a number. There's at least 5 of them. Blood is going to transport dissolved gases like oxygen and carbon dioxide. And that one might be obvious, but also the blood is going to be essential for moving nutrients and hormones around the body and for collecting metabolic waste and delivering it to, say, the kidneys or the liver, wherever it needs to go. The blood is also going to play an important role in regulating pH and the ion composition of interstitial fluids. Keep in mind, and we're going to talk a little bit more about this, but blood, one major component is plasma. That's kind of the fluidy part of the blood. And blood plasma and the interstitial fluid, that's the fluid that surrounds your cells out in the tissues, they are very similar. And the pH of blood is typically about 7.4. And we really cannot tolerate swings of pH beyond, say, 7.35 to 7.45. So it is a very narrow homeostatic range. Blood is also important for restricting fluid losses at injury sites because the clotting process is going to start with platelets that are found in the blood. And we'll talk about that towards the end of this presentation. The blood also, of course, has white blood cells in it, and that is going to help us defend against toxins and pathogens. And also, the blood is very good at absorbing heat from core places in your body like muscles or organs and then redistributing that heat out around the body. And if necessary, maybe we're too hot, then capillary networks close to the surface in the skin can help release that heat, or we can restrict flow into those areas and help hold on to that heat. So a lot of really important functions. A good time to also remind ourselves that when we think about the cardiovascular system, and this presentation is focusing on the blood, but the cardiovascular system doesn't just include the blood. It is also going to include the heart, which we will address in another presentation and lecture, and also blood vessels. So 3 main components of that cardiovascular system. All right, so let's remind ourselves about the components of blood. We are going to have 55% roughly of whole blood being plasma. That's, like I mentioned, the kind of fluidy or liquid part of the blood. It's going to contain plasma proteins like globulins and fibrinogen. It's going to include lots of water. Then we're going to have the remaining part be the formed elements. We don't call them formed cells because platelets are not whole cells. And so formed elements is a better name. So they're going to represent about 45% of the whole blood, and that will include the platelets, the white blood cells, and the red blood cells. And we'll talk about all of these in a little more detail. In terms of big picture characteristics, blood is typically around 100.4 degrees Fahrenheit, and there's going to be a little swing there individually. It does have a high viscosity. That means it's much thicker than water. Think more, like maple syrup or molasses than water. And the volume is going to be a little larger in males, 5 to 6 liters on average versus 4 to 5 liters in females. So just a good overview. Now, digging in a little bit more to the components of these, let's focus on plasma first. So again, here are these plasma proteins, other solutes, and water that we saw on the previous side. Note that water is 92% of the plasma. But let's take a peek at these plasma proteins that make up about 7%. So you can see we have albumins. These are going to be important because they shouldn't be present in urine if your kidneys are healthy and working normally. So by looking for albumins in urine, we can get a quick assessment of what's going on at the kidney level. And if we do see albumins, that is a concern for kidney function. It may indicate something like diabetes, for example. or if we find glucose in the blood, which is, in the urine, which is something else we shouldn't find in urine typically. We may also, we will, have globulins. These are not something we're going to talk a whole lot about, but these are going to be important and connected to the immune system, so they will come up later in the course. And then fibrinogen is going to be important for us in this unit because it's going to play an important role in the clotting cascade. Also note that down here we do have solutes that make up a small percentage of the plasma. These are going to include salts, which we call electrolytes. That would include things like sodium and chloride and calcium, all of which are going to be important signaling molecules, for example, and also organic nutrients and wastes. Then over here in the formed elements, this should look familiar, I think, from lab. We're going to see platelets. They occupy a very small amount of the formed elements, less than 0.1%, and they're sort of just little bits of cellular material from megakaryocytes. And then we're going to have the white blood cells. We've got these 5 different kinds here. Again, they are making up a very tiny percentage of our formed elements. We're going to have those neutrophils, eosinophils, and basophils. All of them will have granules, and then we'll have the lymphocytes and monocytes as well. And of course, the bulk of the formed elements will be the red blood cells. No nucleus here, unlike our white blood cells that have really kind of obvious nuclei. Our red blood cells are lacking nuclei, most organelles. They are very specifically designed for their function, and we'll talk a little bit more about that. Also, some key terms associated with the formed elements are the term hematocrit. That is where we measure the packed cell volume, or what percent of formed elements is in the whole blood. So you can see we have a range of 37 to 54% with that average of 45%. And it will be a little higher. So in males, that range may top out at 54%, but in females, that range may not go as high as 54%. And that is mostly due to the presence of testosterone, which can stimulate blood cell production. All right, so we have a fancy word called hemopoiesis, which is the formation of formed elements in the red bone marrow. It can also be called hematopoiesis, so just be aware of that. We got multiple words going on there. All of our cells, all of our blood cells are going to begin with stem cells known as hemocytoblasts. Those hemocytoblasts will split via mitosis into either lymphoid stem cells or myeloid stem cells. Now, the lymphoid stem cells will go on to become lymphocytes. So once you have a lymphoid stem cell, there is only one end in sight, which is becoming a lymphocyte. But for the other stem cells called myeloid stem cells, there is a greater variety of where these cells could end up. So from myeloid stem cells, we will form progenitor cells, and then those progenitor cells will ultimately differentiate into one of these 4 types of cells: monoblasts, which will become monocytes, or myeloblasts, which will ultimately become either neutrophils, eosinophils, or basophils. Megakaryocytes, which will ultimately form platelets, and of course, proerythroblasts will ultimately become red blood cells. So let's just take a look at this great diagram that's in your textbook. So here we are in the red bone marrow, where we have our hemocytoblast stem cells. And again, they can divide using mitosis to form either lymphoid stem cells or myeloid stem cells. That will depend on the chemical signaling that they receive. The lymphoid stem cells will ultimately form lymphocytes. On the other hand, the myeloid stem cells, they can go on to either become monoblasts, myeloblasts, or proerythroblasts. If they become monoblasts, they'll move through stages until they become monocytes. If they become myeloblasts, they will become one of these granulocytes like the neutrophil, the eosinophil, or the basophil. So here are our white blood cells up here, coming from 2 different stem cells, right? So the lymphocyte from lymphoid stem cells, all of the others coming from our myeloid stem cells, which became either monoblasts or myeloblasts. Now, we also could have that myeloid stem cell go through the progenitor cell stage and become a megakaryocyte, in which case it'll hang out in the red bone marrow and give rise to platelets. And then the last one, and maybe the most interesting, that's maybe unfair, they're all interesting, but we also have the option that the myeloid stem cell, due to stimulation from erythropoietin or EPO, will become a pro-erythroblast. This pro-erythroblast will ultimately eject its nucleus, becoming a reticulocyte, which is basically like an immature red blood cell. And you can find these in the blood. And then they will very quickly form and become red blood cells, which officially are erythrocytes, circulating around in the blood, carrying oxygen around the body to all of our cells. So this is an important concept here, how these blood cells develop. All right, so let's talk a little bit more about red blood cell formation and the role of erythropoietin in that formation. So EPO is released in response to anemia, which would include or might be detected by things like a decreased blood flow to the kidneys, decreased oxygen in the lungs, for example, from being at high altitude where the concentration of oxygen is lower, or due to damaged respiratory surfaces. So any of these things means that we're going to have less oxygen, and that is going to stimulate the release of EPO. And EPO will travel to the red bone marrow where it will stimulate stem cells to produce ultimately red blood cells, because by increasing the number of red blood cells, that leads to an increase in hemoglobin in the blood, and more hemoglobin, because you have more red blood cells, means you can carry more oxygen and deliver it to the cells of your body. Now, in terms of platelets, these are interesting little elements. They are their maintenance, or really a better wording might be what they do. Their function in homeostasis is managed through positive feedback loops, which are the rarer type. Most homeostasis is managed through negative feedback loops. So again, just remind ourselves, this is where our hemocytoblasts have divided to form myeloid stem cells, which then go on to become megakaryocytes, which will give rise to platelets. And what they look like in a blood smear are little flattened discs of, I would argue that they kind of have an unusual shape. In the blood smear, you can see that their abundance is about 350,000 per microliter, but there's a pretty large range here from person to person. What they do is they clump together and they stick to blood vessel walls, which can help heal tears or injuries to the wall of vessels and keep the blood inside the vessel. So in other words, they are very important in the clotting process. Platelets are continuously replaced. Each platelet only circulates for a week to a week and a half, and before phagocytes remove them, and that is going to happen mostly in your spleen. So that's the platelet. And then we can also think about blood in terms of hematology. So don't mix up hematology with hemopoiesis or hematopoiesis. Hematology is the study of blood and blood-forming tissues. So fancy word here, dyscrasias are any blood disorders. And of course, because blood is so essential to the health of all of your cells and tissues, typically dyscrasias will have an effect systemically. So they affect your whole body if you have one of these. So blood tests are important. One thing we can do with a blood test is we can determine blood type, which we'll talk about in a little bit. We can do a complete blood count, so we can look to find out how many RBCs do we have, how many white blood cells, and how many platelets do we have in 1 microliter. This is something that we did in lab, so this should be a little bit familiar. And of course, we could also do a differential count of the white blood cells. So instead of just being like, oh, we have this many white blood cells in total, we could actually figure out how many of each white blood cell we have. And we typically look at those as percents. So over here on the right, we have this complete blood count. This is sort of what maybe a normal blood test might reveal. So you can see here's our hematocrit, or the packed cell volume, at 42%. So the whole blood was spun or centrifuged and fractionated into its parts. So 42%, which would be a normal range. Our hemoglobin, we also, if we look down here, we have our platelet count. This looks normal. We've got our RBCs also looking good. Our total white blood cell count at 7,000 per microliter. But if we want to know more, we would need a white blood cell differential count. And in this case, it reveals we've got about 67% of those white blood cells are neutrophils, 25% lymphocytes, 3% monocytes, 4% eosinophils, and 1% basophils. And those all make sense for what we would typically expect. If we pop over in your book, I think this is on page 637 for you, and it's much prettier looking than the one I have here. But this is a great chart that will sort of give you a little bit more information about each of those tests, and then also the terms that we associated with either elevated or depressed values. So for example, with the hematocrit, we know already that we're measuring the percent of formed elements in the whole blood. And if the hematocrit is high, that could be connected to polycythemia, or if it's low, it may be anemia. We might not know the cause. Why do we have that high or low? But we can describe it. We can look at hemoglobin. This is going to be very similar to the hematocrit. We don't really go into mean corpuscular hemoglobin or mean corpuscular volume, but just take note that mean corpuscular hemoglobin, right? The more hemoglobin you have, the more oxygen you can carry, and the less you have, the less oxygen. But down here, we've got our RBC count. So if our RBC count is high, that would also mean our hematocrit was high. Hopefully that makes sense to you. That would indicate erythrocytosis, high levels of RBCs, or polycythemia. Again, it's not telling us the why, it's just how we describe it. And then if our red blood cells are low, we would call that anemia. We might not know why we have anemia, but we know that we have it because our red blood cell count is depressed. We could also look at the number of immature red blood cells, or reticulocytes. And if we have too many, that would be reticulocytosis. And on the other hand, if it's not the normal amount, it's a little lower, we would call that diminished erythropoiesis. So this is a good little chart just to make sure you're putting all these pieces together from lab and lecture. All right, so let's go a little more into these red blood cells. So this might look familiar. You saw something similar in lab when you were looking at a blood smear. The overwhelming cells that you see in a blood smear are the red blood cells. They have no nucleus. There are a lot of them, up to 6.3 million per microliter. They are about a third of all of the cells in your body. And they have a very unique structure or anatomy that is perfect for their function. So they have a very large surface area to volume ratio. In other words, their volume is small relative to how large their surface area is. And that's in part due to this indentation that kind of gives them a donut shape, and that increases the surface area. And the more surface area there is, the easier it is for oxygen to diffuse across and bind to hemoglobin inside. Red blood cells can also form these really cool little stacks because of their shape. Those stacks are called rouleaux, and that allows them to move through tiny little capillaries to deliver that oxygen out to your tissues and cells. They are also quite flexible, so they can kind of bend in the middle, which again lets them fit through tight capillaries. And they have no membrane-bound organelles. So all those things you learned in AP1 way back when, all those organelles, they don't have any of those membrane-bound organelles. They are anucleate, no nucleus, because they are filled with hemoglobin. They've got this one job, and they are designed to carry out that one job of holding on to oxygen and moving it around the body and delivering it. Now, hemoglobin itself is kind of interesting. So, again, a little flashback to A&P1 lecture. You should have learned about protein structure at the primary, secondary, tertiary, and quaternary levels. So, hemoglobin is a quaternary structure protein. That means it has all 4 levels of protein structure. So, it has 2 alpha and 2 beta chains, and they are built independently by protein synthesis at the ribosome and folded in the rough endoplasmic reticulum. But ultimately, these polypeptides come together into this quaternary complex structure to form hemoglobin. Inside the hemoglobin is something called heme, which is a pigment that holds iron. And because of that, it can hold O2. And when oxygen binds to iron on the heme, we call it oxyhemoglobin, and it does change color, and that's connected to the fact that it's a pigment. You have about 280 million hemoglobin molecules in each red blood cell. I mean, there's just so many because that's how important oxygen is. But here's what's really fascinating. Carbon monoxide actually binds better to hemoglobin than oxygen does. And the reason why is that when hemoglobin evolved, there was a very high concentration of oxygen in the atmosphere and almost no carbon monoxide. So it didn't need that flaw, if you will, wasn't an issue because there wasn't any carbon monoxide to bind. But the consequence of hemoglobin evolving through a very rich oxygen environment is that nothing eliminated the fact that it could bind carbon monoxide. So now, if our hemoglobin is exposed to carbon monoxide, it will preferentially bind the carbon monoxide over oxygen, and then oxygen doesn't get delivered to our cells, including our brain cells, and that means they will quickly be unable to function and maintain what they do to keep us alive. So that's why any kind of carbon monoxide is quite dangerous. Okay, so how are red blood cells produced and then eliminated? Because we are replacing about 1% of our red blood cells every single day, and they circulate for about 3 months. So this is a little bit of a complicated process, but I think this diagram kind of makes it not so bad. So let me just see if I can make it a little bit bigger. All right, so here we go. Let's just start right here at number 1, right? So we kind of already know this part. We talked about it. We're in the red bone marrow, and pro-erythroblasts that are coming from stem cells will go through the stages of becoming reticulocytes, which will ultimately become red blood cells. A few important things have to happen during this early stage of development. So you'll notice that these very early red blood cells or developing red blood cells are going to absorb iron from the bloodstream so that they can synthesize hemoglobin molecules, because of course hemoglobin is going to be super important for their ultimate function when they're all grown up. So once we get these reticulocytes that can go out into the blood, and they will account for less than 1% of circulating red blood cells, and within 24 hours, those reticulocytes will complete their maturation and they will resemble all the other red blood cells that are circulating around. So here we are, and notice that their lifespan is about 120 days. At some point, one of sort of 2 things is likely to happen. So let's bring it over to here. So they may just start to disintegrate in a process called hemolysis in the bloodstream, and in which case they will get picked up by macrophages. Sometimes macrophages will actually just engulf whole red blood cells that are old and break them down. But either way, macrophages in the spleen, in the liver, and in the bone marrow are going to be collecting old red blood cells. And the reason why is because they want to grab the heme and remove the iron from it to send the iron over to the red bone marrow for the new red blood cells that are developing. Because that iron is hard to come by in our diets, and we need that iron attached to heme in the hemoglobin in order for us to carry oxygen effectively. Now, once we've removed the iron from the hemi, that hemi is converted to biliverdin, which becomes bilirubin, and then bilirubin actually binds to albumin in the bloodstream, and eventually that will end up in the liver, and the liver is going to excrete that bilirubin in bile into the intestines, and that's how we'll get rid of all of that waste there. But notice, just a reminder, we did not get rid of the iron. The iron we are recycling because that's important. Another step that's happening, just to remind ourselves, is we also have events that are occurring in the kidney. So some hemoglobin and urobilins, which come from the intestines, from the bilirubin that was secreted, we may reabsorb some of it in the form of urobilins. These can also be picked up by the kidneys and eliminated in the urine. So one way or the other, we are eliminating these waste products. So big picture here. I mean, there's a lot of details, but let's focus on the big picture. Red blood cells develop in the red bone marrow. In order to develop well, they need iron. So your body has a system for recycling the iron from old red blood cells by having macrophages break them down and recycle the iron back to the bone marrow. And then the rest of the waste and hemi will be eliminated through bilirubin in the liver, and any other odd bits will be excreted in the urine. So that's the big picture there. All right, that brings us to blood type. I'm not going to spend a lot of time on this because this is something we hit in lab, and so you should be pretty familiar with this. But just want to remind you that red blood cells may display antigens on their surface. They might have type A antigen, type B antigens, or sometimes both, and sometimes they display no antigens. What antigens are displayed by red blood cells is determined by genes that are passed from parent to offspring. People with type A have never seen antigen B, and so they will have anti-B antibodies in their blood, and the opposite will be true for type B. They will have anti-A. AB won't have any antibodies, and type O, since they don't have either of the antigens, they will have antibodies to both. And this is going to be important in terms of what blood can be transfused to other people. So we want to make sure that we give people blood that will not trigger an immune response. So if someone is type A, we don't want to give them type B because they will have type B antibodies and they will attack the blood that's being transfused, resulting in clumping, and that can lead to death of the patient because the vessels are going to become clogged as a result of that agglutination or clumping. So here's the overview there. So what we can do to determine blood type, if we have a mystery blood sample, if we put anti-A antibodies and nothing happens, then we know that there's no A antigen on that blood, because if there was, it would have clumped, right? So when you do blood typing, whatever clumps, that's the positive result. So if it's type A blood and we add anti-A, it will clump, and that tells us that's type A blood. If it's type B blood and we add anti-B antibodies, then it will clump, which tells us that's type B. We also have to remember, if there's no clumping from anything, then it's type O. If it clumps with both A and B antibodies, then we know it's type AB blood. So the other thing we have to remember is there is that Rh factor. And so we also have to test for that, because if someone is Rh negative, we cannot give them Rh positive blood because they will have antibodies to that Rh factor and it will agglutinate or clump. And that's never a good thing. We don't want that. So this is a good little chart to just confirm what you already learned in lab in preparation for our unit test as well. This is just an interesting chart from your book. You definitely do not need to memorize this for our test, but I just think it's really interesting to see how different groups of people have different, are more, there's like different percentages of people with each of the different blood types. So if you're interested, like in AB, we see the percentage of AB blood types is highest in Japanese and Korean Americans. Whereas if you look at, say, type B blood, that's really high among like Chinese Americans and Filipino Americans, and again, Korean Americans. But then like type O blood is really high among Black Americans and also Caucasian Americans, and not so high among some other groups. But here we can see in the Hispanic population, like 57% have type O blood, which means there's way fewer of type A and type B. And if you look at type B, for example, less than, only 10% or less have type B. So this is going to be the rare blood type there. On the other hand, among some other groups, it's pretty high. So there's just some interesting things there. And you can see that for AB blood type, this is pretty low in most groups. So just if that interests you, you can take a minute and check that out. All right. Also in lab, you learned about hemolytic disease. This is the reaction between an Rh negative mother and an Rh positive baby. We have essentially eliminated this issue in the United States by using a vaccine called RhoGAM. So the issue is that if mom is Rh negative, then she will form antibodies in response to Rh positive factor if her immune system sees it, so to speak. So if dad is Rh positive and baby inherits the gene from dad, mom's immune system may be sensitized during labor and delivery. And as a result, she will produce antibodies. Then if she gets pregnant again and the second baby is also Rh positive, mom now has antibodies against that Rh positive factor, and those antibodies can cross the placenta. That's kind of interesting because the AB antibodies are too big and they can't get across, but the Rh ones can. So if mom has Rh antibodies and the baby is Rh positive, then mom's immune system will destroy the fetal red blood cells because of that presence of that Rh factor. And that leads to a high mortality rate for the developing embryo or fetus. So we want to prevent that, and we can do that by giving mom the RhoGAM vaccine during the first pregnancy if mom is Rh negative, in order to make sure that mom doesn't produce anti-Rh antibodies. And that way, in a second pregnancy, we don't have to worry that mom's antibodies attack an Rh positive fetus. So it's just another great example of how much vaccines have positively changed patient outcomes in the United States and around the world. So that brings us to white blood cells. So these are also known as leukocytes. They are very different from red blood cells because they have organelles and they do not have hemoglobin. You've already learned quite a bit about these in lab, but let's just point out the shared properties among the leukocytes. They almost all can be found in both loose and dense connective tissues. They actually only spend a short time in the blood. That's mostly just how they move around. And then they are able to squeeze between the endothelial cells to get out into the surrounding tissues. And that process is actually really unique. Most cells can't do it, so it's kind of unique. And we have a fancy word called diapedesis. And you might see the PED in there, which is like pedestrian or walking. So the white blood cells can walk between the vessels to get out, between the endothelial cells of the vessels to get out into the connective tissue. They also exhibit positive chemotaxis. That means that many of them will respond to chemical stimuli to pull them in, for example, towards a site of injury or infection. And most of them, not all, but most of them are capable of phagocytosis. So that's really important too. And just a reminder, we can take the leukocytes and split them up into the granular leukocytes and the agranular. Our agranular leukocytes are going to be the monocytes and lymphocytes, and then our granular leukocytes will include the neutrophils, the eosinophils, and the basophils. And remember, when we looked at them under the microscope, they all had really obvious granules in them, and they are also pretty hard to find in a blood smear because there aren't actually that many of them in any given blood smear. Okay, in your book, there is a prettier version of this chart, but I've put it here because I think it's pretty helpful. So on the right side, we have sort of a cartoon image of each of these cell types just to remind you what they look like and how you can recognize them. And then in our chart, this will go through each one of these and give you a basic description of what they look like. I think this is good to review. So this column and the last column should go together. And then also you should know, you need to know their basic functions. And we covered this in lab as well. So we know that neutrophils play a really important role in bacterial infections by engulfing pathogens and destroying them. Eosinophils play a really important role in parasitic infections and allergens. They also may help reduce inflammation. Basophils are going to release histamine and other chemicals that promote inflammation, which, while we always think of inflammation as negative, it does play an essential role in tissue injury and healing. Monocytes, of course, are going to be really important in phagocytosing pathogens and other debris. And then, of course, our lymphocytes are going to help provide defense against specific pathogens or toxins. So they are going to be targeting something that is, present in our system, and we want to go after that particular pathogen. So a good chart to spend a little time with. Okay, that brings us to clotting, because you can't talk about blood without talking about clotting. Clotting happens in 3 phases: the vascular phase, the platelet phase, and the coagulation phase. And I do always like to point out that the fancy name for the clotting response is hemostasis, which is awfully close to homeostasis. So just make sure when you're taking a test, make sure you double check if the question is asking about homeostasis or hemostasis. They may be related because hemostasis is essential to homeostasis, but it could definitely lead you down the wrong road if you misread it. So just watch out for that. So that first phase is the vascular phase. We're talking within 30 minutes after injury. We've got lots of important events happening during this phase. We are going to have endothelial cells contracting. They may even expose the underlying basement membrane to the bloodstream, and those endothelial cells will begin releasing chemical factors and local hormones. That will include things like endothelins, which are a peptide hormone that stimulates smooth muscle contraction. So that will help close down the vessel and help prevent the loss of blood through the injury. And it will also stimulate fibroblasts to accelerate the repair process. And then another little thing that happens in the vascular phase is that the membrane becomes kind of sticky. And that's really important because that means that the endothelial cells on opposite sides of the injury will actually stick together to help kind of temporarily close the damaged vessel. And then also it allows platelets to attach to the damage. And as platelets attach, that's going to then induce the platelet phase. And in the platelet phase, we're going to see platelets really sticking to those sticky endothelial surfaces, and they will stick to each other and collagen fibers in the area, and they will start secreting lots of chemical signals, which in turn will bring in more platelets. And so they will really clot up in that area. And that is going to include things like platelet factors and platelet-derived growth factor. And we will even have calcium ions because these are going to be needed for platelet aggregation and for the clotting process, which we're going to go into in a little bit more detail. And then the last part is going to be the actual coagulation phase. And let's flip to our Sorry, I got a little ahead of myself here. This is an image of the platelet phase. So you can see that the platelets are starting to attach to the endothelial cells. And again, we have the platelet factors, PDGF, and calcium all happening here to help bring in more and more platelets so that we can get to the actual coagulation phase, which looks a little scary and it is important. You do want to spend some time on this. But basically, there are 3 cascades, all right? And the point of all of these cascades is to get fibrinogen to fibrin, because fibrin is what we need to start forming a really good clot. And so, because this is so important, your body has evolved 3 ways of activating factor X, because as long as we activate factor X, we can convert fibrinogen to fibrin. And if we can do that, we can begin this coagulation phase. So in the extrinsic pathway, that's over here, we are going to have from cells in the damaged area, we're going to have factor 3 that along with calcium will form factor 7, which will lead to a tissue factor complex that activates factor X. So let's just sum that up in way shorter terms. Endothelial cells at the site of injury will release factor 3, which will ultimately result in the activation of factor X. That's cascade 1, extrinsic. On the intrinsic side, we have a whole lot of things going on, but again, we are going to have factors released from platelets, and along with calcium, we're going to have, in combination with factors 8 and 9, that will lead to the formation of the factor 10 activator complex, which will result in the activation of factor X. So we have a second pathway that, due to chemical signals being released from the injury site, we activate factor X. That's the intrinsic pathway. And then the final pathway is the common pathway. This is where factor X has been activated. Factor X becomes prothrombin. Prothrombin turns into thrombin, and thrombin is what converts fibrinogen that's normally in the blood. That's one of the proteins way back and early on in this lecture in the plasma that will then be converted into fibrin. And everything sticks to fibrin. So if we get lots of these little fibers, it creates almost like a little mesh network, and all of this stuff can stick to it and form a clot to close up this tear in the vessel. Now, this isn't going to work in like an aortic tear, right? These are relatively small vessel tears and injuries that this will work on. Ultimately, the last thing is the clot will actually retract and we'll get this vessel working again. But this is the process. So yes, you need to know this, but focus on the big picture. Both the extrinsic pathway and the intrinsic pathway lead to the activation of factor X. Factor X ultimately gives us prothrombin, which becomes thrombin, which converts fibrinogen to fibrin, which leads to coagulation. All right. So, start with the big picture before you bog down in the details. All right, let's end with talking about some blood disorders. Some blood disorders may be nutritional blood disorders. That would include iron deficiency anemia or pernicious anemia. So iron deficiency anemia is when you have normal hemoglobin synthesis not occurring because there aren't enough iron reserves or you're not eating enough iron. And as a result, the red blood cells are very small, which is called microcytic because they don't have enough hemoglobin in them. This is much more common in women because they are much more reliant on a dietary intake of iron because women's iron reserves are not as robust as men's. So it's really important to make sure you have enough iron in your diet. Pernicious anemia is connected to a deficiency in vitamin B12. Vitamin B12 is really important for normal cell division of stem cells in the red bone marrow. So without B12, we can't form those essential myeloblasts to form red blood cells. And then what will happen is our red blood cells become very large or macrocytic. Another issue are blood infections. So septicemia is when you have pathogens that are present and multiplying in the blood and spreading throughout the body. It can definitely be fatal, and it is difficult to treat if we don't get an early start on it. We also have congenital blood disorders like sickle cell anemia, thalassemias, and hemophilia. So, hemophilia is when there is an issue in any one of those clotting factors. So, just real quick, I'm going to pop back here. Remember, in here, we've got factor 8, factor 9, factor 3, factor 7, factor 10, right? So there's different kinds of hemophilia. Does it make sense that if the problem is in factor 10, that's a problem? Because that means you have no way to get prothrombin to thrombin to convert fibrinogen to fibrin. So a problem here is a really big problem. A problem over here in factor 8 or 9 or in 3 or 7 would only impact one of the pathways, either the extrinsic or intrinsic, which means that individual would have the ability to clot, but not as easily and not as quickly as someone that has all functioning factors. So there are different genes for each factor, and depending on which factor is compromised, it will have a greater effect. Again, factor 10 being the really big one here. Now, sickle cell is an interesting one. You've probably learned about it in high school, but sickle cell is due to a defect in the gene for hemoglobin, which results in the hemoglobin not having a normal quaternary structure, and so the shape becomes sickle-shaped, and that makes it really hard for the cells to hold oxygen and to move through blood vessels. So they can get stuck inside capillaries, which can cause pain, and patients can be exhausted because their cells are not getting enough oxygen to make enough ATP to meet the metabolic demands. Thalassemia is actually in the same family as sickle cell, and it's somewhat similar. And something that's kind of interesting about sickle cell is we see it among people that live in areas where malaria is also epidemic, because having sickle cell protects you from malaria, particularly during your younger years. So if you have sickle cell, you're less likely to die of malaria as a child, and you're more likely to live long enough to have a child and pass on your genes. So from an evolutionary standpoint, that's a win. But the problem is sickle cell without treatment leads to death, I don't know, in your 30s, maybe 20s or 30s. But that's better than dying as an infant who gets bit by a mosquito while in their crib and dies from malaria as an infant. So, evolution's a funny monster, right? So those two diseases, evolutionarily maybe go together. We do have lots of great treatments for sickle cell today, including the newest being CRISPR-Cas9 technology, where we actually go in and fix the defective gene in stem cells in the bone marrow. And so patients can then start making normal hemoglobin, which means we're essentially curing the disease. So they've had a lot of success with this in the first, it's only been going for maybe 5 years or so. Don't quote me on that. But it looks really good. And hopefully we're going to see even more of that, because it will be great for patients. And then there are also blood cell cancers like leukemias. Those are going to affect like the myeloid leukemia will affect the granulocytes like neutrophils, eosinophils, and basophils, whereas lymphoid leukemias will, of course, affect the lymphocytes. And leukemias have to be treated for patient survival. So these can be tough cancers to treat, but again, we've made big headways in treating some of these kinds of blood cell cancers. Your book also mentions degenerative blood disorders like disseminated intravascular coagulation, which can be caused by bacterial toxins that activate steps in the coagulation process, converting fibrinogen to fibrin when we don't need it. And so it creates clots in the vessels. And obviously, that's not great, because We want blood to be able to move freely around the body to deliver oxygen. Okay, so you can also visit all of these blood disorders at the end of your chapter if you want to learn a little bit more about them.